Chapter 20, the heart. The pulmonary circuit carries deoxygenated blood to the gas exchange surface. The systemic circuit carries oxygenated blood to the rest of the body. Circulatory components. Arteries carry blood away from the heart. Veins carry blood toward the heart. Capillaries are thin vessels that connect veins and arteries. They allow also for gas and nutrient exchange. Heart anatomy. Pericardium. The pericardium is the lining of the pericardial cavity. The visceral pericardium, also known as the epicardium, covers the surface of the heart and is a single cell layer. The parietal pericardium covers the surface of the pericardial sac, or the outer layer. The pericardial cavity is the space between the visceral and parietal pericardium. This cavity contains pericardial fluid, which is a lubricant. The heart wall. The epicardium, also known as the visceral pericardium, is made of mesothelium and connective tissue, and more specifically, it's made of simple squamous cells attached to areolar tissue. The myocardium is muscle, nerves, and blood vessels, and it does not necessarily need the CNS in order to work. The endocardium is endothelium and connective tissue. It lines the inner chambers of the heart, i.e. the atria and the ventricles. Cardiac muscle tissue. The cells are small, branched, striated, and mononucleated. The cells join at intercalated discs with gap junctions, which is a direct synapse and not a chemical synapse. The striations in cardiac tissue run parallel to intercalated discs. Superficial anatomy. The base is the superior end and the apex is the pointed inferior end. The auricles are the extensions of the atria and are the upper chambers. The atrioventricular sulcus, also known as the coronary sulcus, is the groove between atria and ventricles. It does not separate the two. The interventricular sulcus is the groove between the ventricles, and it has an anterior and posterior aspect. Heart chambers. The right atrium receives deoxygenated blood from the systemic circuit via the superior and inferior vena cava. The right ventricle receives deoxygenated blood from the right atrium and pumps deoxygenated blood into the pulmonary circuit via pulmonary arteries at a lower pressure because it's only pumping to the lungs and therefore it's easier to pump. The right ventricle also contains trabeculae carnae, which are the topographical ridges. The tricuspid valve has three cusps. The chordae tendineae attach cusps to papillary muscle. The tendineae keep blood from dumping back into the right atrium. Pulmonary semilunar valve. This has three cusps and guards the pulmonary trunk and arteries. It has no chordae tendineae. The left atrium receives oxygenated blood from the pulmonary circuit via pulmonary veins. The left ventricle receives oxygenated blood from the left atrium. It pumps oxygenated blood into the systemic circuit at a higher pressure, and that is partially due to the fact that it is thicker muscle. The bicuspid valve has two cusps and chordae tendineae, which attach cusps to the papillary muscle. The aortic semilunar valve has three cusps, no chordae tendineae, and guards the aorta more like a doorway. Coronary vessels. Coronary arteries. Coronary arteries supply oxygenated blood to the atrium and the ventricles. Blood enters by aortic elastic rebound during the diastolic phase. Coronary veins. Coronary veins drain deoxygenated blood from the heart. Cardiac cell types. Conducting cells control and coordinate heartbeat as action potential moves through the conducting system. Contractile cells are the cells that produce contractions and propel blood. The conducting system. The conducting system coordinates heartbeat. Sinoatrial node. The sinoatrial node has pacemaker cells which establish heart rate. They are found within the right atrium. Internodal pathways. Internodal pathways connect the SA and the AV nodes. They pass action potentials to the atrial contractile cells. Atrioventricular node. The AV node is where the action potential is delayed. The atria contract. It is found in the floor of the right atrium. Atrioventricular bundle, also known as the bundle of His. This is what moves action potentials down the interventricular septum. Bundle branches. These move the action potential down the interventricular septum. The action potential moves to the moderator band. Papillary muscle contracts, chordae tendinae tense, and they keep the cusps from flipping back into the atria. This keeps the valves from backflowing. Purkinje fibers. This is where action potential moves across the ventricles. Contraction from apex to base that directs blood upwards. Fiber goes straight down to apex, then fans upward. Contractile cells are the cells that produce contractions. The next few points are regarding the action potential of contractile cells. Depolarization. Sodium channels open fast and sodium moves in. Plateau. Sodium channels close fast. Calcium channels open slow. Calcium moves in while sodium is pumped out. Plateau determines how rapidly ions can move in. Repolarization. Calcium channels close slow, potassium channels open slow, and potassium moves out. Also in repolarization, the transmembrane potential is restored and potassium channels close slow. The next 2 points are about the refractory period. The absolute refractory period is when the membrane cannot respond to a second stimulus. The relative refractory period is when the membrane can respond to a greater than normal second stimulus. Electrocardiogram, also known as ECG or EKG. This test monitors the electrical activity of the heart. P wave. The P wave corresponds to atrial depolarization, and the atria contract after P peak. QRS complex. The QRS complex corresponds to ventricular depolarization. The ventricles contract after R peak. Atrial repolarization occurs in the QRS complex, but it is masked in an EKG. T wave. The T wave corresponds to ventricular repolarization. It infers that the ventricular muscle is relaxing. PR interval. The PR interval is from the start of atrial depolarization to the start of ventricular depolarization. QT interval. The QT interval is from the start of atrial depolarization to the start of ventricular depolarization. In atriosystole, the atria contract and top off the ventricles with blood. In atrial diastole, the atria relax. This next point deals with ventricular systole. The ventricles contract. The pressure inside the ventricles is greater than the pressure inside the atria. The AV valves close. Then there is greater pressure in the ventricles than in the aorta or pulmonary trunk, and thus the semilunar valves open. When this occurs, blood fills the aorta or pulmonary trunk. There is more pressure needed in the ventricles to close the valves. Ventricular diastole. In ventricular diastole, the pressure inside the ventricles is less than the pressure in the aorta and pulmonary trunk. Semilunar valves then close. Then the pressure in the ventricles is less than the pressure in the atria, and thus the AV valves open. When this occurs, there is a passive filling of the ventricles. Also in ventricular diastole, the heart is relaxed, and there is more pressure in the atria, which push the valves open, and the ventricles fill with blood. Heart sounds. A stethoscope is used to hear heart sounds. The first heart sound, S1 or lub, is the closing of the AV valves. It also marks the start of ventricular systole. In the second heart sound, S2, also known as dub or dup, the sound is made by the semilunar valves closing. This marks the start of ventricular diastole, or relaxation. Cardiodynamics. Cardiodynamics are the movements and forces during the cardiac cycle. End diastolic volume, EDV. The end diastolic volume is the blood volume in each ventricle at the end of ventricular diastole. End systolic volume, ESV. This is the blood volume in each ventricle at the end of ventricular systole. Stroke volume. The stroke volume is the blood volume pumped from each ventricle in one heartbeat. The equation for stroke volume, also known as SV, is the end diastolic volume minus the end systolic volume. Cardiac output, CO. This is the blood volume pumped from each ventricle in 1 minute. The equation for cardiac output is the heart rate times the stroke volume. Neural control of heart rate. Centers in the medulla receive chemical and pressure information. The SA and the AV nodes are innervated. Acetylcholine opens potassium channels. This slows the depolarization. Norepinephrine opens sodium and calcium channels. This speeds depolarization. The brain registers how much CO2 levels are in the blood and responds appropriately. The brain does not register the level of O2.